Okay, so moving on, we've looked at R groups. Now, these R groups can basically be saturated and unsaturated. Now, these are two really key terms that you need to be familiar with. Okay, now if an R group is saturated, think of the R group being normal. Okay, that's basically what happens when the R group is saturated. Saturated means that it's normal. Unsaturated means that it's not normal because it has this dodgy un before it. Unsaturated means that it's not normal. When you have a saturated fatty acids, now if you do chemistry, this will make more sense, but there are only single bonds present between the carbons, and therefore it can hold the maximum number of hydrogen atoms. So think about it like this. I've drawn an example over here. This is a saturated fatty acid. This is a saturated carbon chain, and that's because there are only single bonds present. Okay, there are only single bonds present within this sort of molecule. And that's a really important point that you need to be aware of. There are only single bonds present. That's why this is a saturated fatty acid. It's normal because there's no dodgy un before it. Over here, there's an un before the name, and that means that it's not normal because there's a double bond present. There's not only just single bonds present, there's also a double bond present, and that makes it a bit weird. Over here, we can see that there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens there. And over here, 1, 2, 3, 4, 5, 6, 7, 8. We can see that there's only 8 hydrogens. So we had 10 hydrogens here, and we only have 8 over here. And that's what makes this unsaturated. There's a double bond, and because of that double bond, it has caused the the fatty acid chain to not hold the maximum number of fatty acids. The maximum number of hydrogens. So bottom line over here, in a saturated fatty acid, you can only see single bonds. Okay, you can only see single bonds between the carbons. And therefore, because there are only these single bonds, that allows this chain to hold the maximum number of hydrogens. Remember, one carbon can bond with 4 other atoms. Now, in an unsaturated fatty acid, there's at least one double bond. Okay, so you can see over here, there is this double bond. And because of that double bond between the carbon atoms, it can't hold the maximum number of hydrogen atoms because this carbon is bonded to this carbon twice, and it can only bond to 2 other hydrogen instead of 4. That's why you have 8 hydrogens over here, whereas you have 10 over here. Okay, so that's the key difference there. And this is a common question. I have an exam question example on this question as well. So make sure you know the difference between saturated and unsaturated. Saturated is normal. There's nothing wrong with the name. Unsaturated, there's a dodgy un before the saturated. That makes it not normal. Therefore, it has a double bond. Okay. Now, finally, the final thing for triglycerides that you need to be aware of are the properties of triglycerides. And we're going to go through these properties one by one. This is a common exam question as well, and people find it difficult to sort of remember. So first of all, properties of triglycerides. They store a lot of energy. Now, triglycerides, remember, it's a one of the groups of lipids. Okay, so it's a group of lipid. Okay, and one of the main sort of properties of triglycerides is that it can store a large amount of energy. Imagine you need to run, you need to respire, you need energy. Where's the energy coming from? It's coming from a storage of fat inside your body. That's one of the sources of energy of where it can come from. So triglycerides store a large amount of energy. Why do they store a large amount of energy? Well, because when we have just C, it's bonded to at least 2 other hydrogen atoms. Okay, and we can make this chain as long as we want. We got CH, CH, and this can go forever. Okay, and as you can see, the ratio of carbon to hydrogen bonds is much larger. There is one over here, 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 one over here. So in that small molecule there, you have six bonds, CH bonds. You have six CH bonds. Because you have six CH bonds, there's only three carbon atoms. Okay, and because there are more bonds, more CH bonds than carbon atoms, each carbon atom is able to hold a large amount of energy. So collectively, for a triglyceride, that provides the large store of energy. That's just a practical way of thinking about it. Ultimately, you just need to know this highlighted bar. Properties of triglycerides, large store of energy. Okay. Second property, it acts as a metabolic water source. Okay, now what does that mean? Well, basically, we know that triglycerides are a group of lipid. So when triglycerides can basically be oxidized, and remember, oxidized means gain of oxygen, or loss of electrons, or loss of hydrogen. But basically, when they are oxidized, they gain oxygen. So when they're used in respiration, they can release water. Why? Well, what does respiration release? Respiration uses glucose and oxygen to produce carbon dioxide and water. And when they produce the carbon dioxide and water, when triglycerides are oxidized, remember triglycerides are a lipid. Just remember that. If you remember the bottom line that triglycerides are lipids, then you're going to remember that they can be used in respiration at some point in the cycle. When they're used in respiration, they can be oxidized to release water. Now, triglycerides, common exam question this, they can be used for animals in the desert. Okay, for example, camels, prime example that they love asking you in an exam. Camels have a large store of triglycerides. Why? Because it comes back to this first point that triglycerides have a large store of energy. And in the desert, where camels, there's less water, they have to conserve the water that they have within their body. And in order to do so, they need to store as many of these triglycerides as they can. Okay, because when they are oxidized, when triglycerides are oxidized, they release water. So camels can use water because there's a shortage of water within the desert. So again, this is a bit of in-depth and application based, but ultimately you need to know this highlighted point over here. They act as a metabolic water source. I'm just explained what that actually means. When they are actually oxidized, what happens is that they release water, and water is needed for people in their daily life. And the reason why this is a common application question is they might put a camel in there, they might put some form of an animal you've never heard of before, but if they do, you know what they're talking about. Camels, they store a lot of triglycerides in their hump, so when they respire, they can provide the water because the triglycerides can be oxidized. Okay, this is a common point over here. The next one, easy to remember. It's the same for starch. It's the same for cellulose. It's the same for glycogen. It's the same for triglycerides. Remember this point. It's not going to change. Triglycerides do not affect the water potential as they are large and insoluble in water. Just learn that point. We've already looked at this point for starch, cellulose, and glycogen. This is a property that you will just know off the top of your head because it's so common in biology and it's a common property that everyone has. It does not affect the water potential because it is large and insoluble. Why does that matter? Well, if you think about it, if it was soluble in water, then it would just dissolve in the water and it would lower the water potential, causing more water to move in. It is large and insoluble, so it doesn't affect the water, the way water is going throughout your body, the way water is moving through membranes in the body. It doesn't affect the way that it's moving by osmosis in the body. It doesn't affect anything really. Okay. And finally, it's relatively low in mass, therefore a lot can be stored. So again, this I've not seen this point mentioned quite often because normally you get the marks with the first 3 points. But if you wanted an additional point, you can have the last one there. So there's a large store of energy, nice and easy one to remember. It acts as a metabolic water source because it can be oxidized to release water. Really helpful for plants, some plants and camels, and does not affect the water potential or osmosis as they are large and insoluble in water. Okay, so those are some common properties that you will be asked about in an exam, so make sure you've made a note of them. Okay, so moving on, phospholipids. That's everything to do with triglycerides. If we have time at the end, we will come back and just review it all again. But if you have filled in the form, you will get the link to the recording to this. Okay, now remember when I said that there are two categories of lipids, there are two groups of lipids. One is triglycerides and the other is phospholipids. Okay, now phospholipids are really important. They actually have a similar structure to triglycerides, which we're going to go through now. This is the structure of a phospholipid. Now you will realize some similarities and some differences within the structure. You have a glycerol, which is the exact same. You have ester bonds, which is the exact same, and you have fatty acid tails, which is the exact same. But the only difference over here is that you have a phosphate. Okay, now you're probably thinking, phosphate, why the hell is that there? Well, I will explain that on the next slide. But the easiest way, it's phospholipid. Phosphor. Phosphor is hinting at something to do with the phosphate. That's why you know that there are not three fatty acid tails. There are two and one phosphate. Okay, so it's made up of one molecule of glycerol. Whenever answering exam questions, make sure you mention the numbers. It's made up of one molecule of glycerol, two fatty acids, and one phosphate group. Okay, and in order for the fatty acids to combine to the glycerol, again, ester bonds have to form. Whenever you're forming a chemical bond, um, bottom line is that it's going to be a condensation reaction. A condensation reaction is going to take place. And over here, because two bonds are being formed, two molecules of water are going to be released. Okay, that's how the two fatty acids combine to the glycerol. Okay, so two ester bonds form within the phosphate group attached to the glycerol. Okay, so we've had a look at the structure of phospholipids over there. Glycerol, two fatty acids, phosphate, two ester bonds releasing two molecules of water, and that's your phospholipid. Okay, similar to the structure of the triglyceride, a bit different because of the phosphate. Now you're probably thinking, what's the point of this phosphate? Why is this phosphate actually there? Well, the phosphate group has a negative charge. Okay, it's not phosphorus, it's phosphate. And if it's phosphate, it's going to be charged. It has a negative charge. Okay, and that's why the glycerol and phosphate group are known as the hydrophilic head. Okay, now what does hydrophilic mean? Hydro means water. So we've got water there. And philic means basically like, so but water loving. Okay, so the phosphate group is basically known as water loving. Okay, because it is charged, it can interact with the water. Okay, and that's a useful property. And I'll show you why. Because phospholipids are actually used in cell membranes. Because they are used in cell membranes, that allows the cell membrane to be partially permeable, to allow some substances through, but not others. And the reason is behind these phospholipids. They have a hydrophilic head. Hydrophilic head is water loving. They can interact with water because they are charged. So if we think about this aside, if we cut this into half like that, this top side over here can interact with the water. Okay, so this top can interact with the water. Okay, however, the bottom, because there are no charged regions at the bottom, what happens over here is that this part cannot interact with the water. This part can interact with water and it can allow water through, and it can sort of mix with water and it can touch water, everything like that. This bottom part cannot interact with water. Okay, so half of it can interact with water and half of it can't interact with water. Okay, now what happens is that because this phosphate group is charged, it repels the other fats. Okay, it has a negative charge on it. So if it is faced with another phosphate, it's going to repel it. Okay, so think about it. We've got one phosphate over here and we've got another phosphate over here. Both of these are negatively charged. What's going to happen is that both of these are going to repel each other. Okay, they're just going to try push into each other, but ultimately they're going to repel each other. Okay, they're not going to have any luck getting close. Okay, so because there's a phosphate group present on sort of every fat, when they come together, they're just going to repel each other because both of them have the same charge. The fatty acid chain, on the other hand, this bottom part is not charged. Because this bottom part is not charged, it's known as the hydrophobic tail. Now, you might be introduced to new words over here, hydrophobic, hydrophilic. But hydro means water. Phobic, if you have a phobia of something, you don't like it. You're scared of it. So hydro means water. Phobic means hating. So the fatty acid chains over here, fatty acid tails over here, they're actually hydrophobic. They don't like the water. The top part loves the water, can interact with water as much as they want. The bottom part hates the water. It doesn't want to interact with water. Yeah, and that's because they're not charged. Because they're not charged, they cannot interact with water. Okay, but again, this can mix with other fats, but it just can't touch water because it's not charged. It can't sort of dissolve and mix with water. Okay, and that's basically phospholipids. Okay, so we've looked at phospholipids. We had a bit of an introduction onto what it has and why it has those components. We're now going to move on to the the properties of phospholipids. If I'm going too fast, guys, please do let me know. And if you do have any questions, please do let me know in the Q&A. I'll be happy to answer them. Properties of phospholipids. First property, it's polar. There are two charge regions, one neutral, one negative. Okay, simple property, it's polar. Second property. Now, this is the property that may seem confusing. It forms the phospholipid bilayer. Okay, and I've got an image of that here. This is what a phospholipid bilayer looks like. Now, if we think about it from scratch, let's draw out a cell. Okay, so we've got a cell over here. Okay, that's the cell membrane, and outside is the cell wall. Okay, we've got the nucleus, we've got some mitochondria, we've got some ribosomes, we've got some chloroplasts. Okay, this is an image of a plant cell. Now, plant cell has a cell membrane. Okay, this is the cell membrane. Now, if we were to zoom in onto the cell membrane using a microscope, what will we actually see if we zoom in? Okay, the structure of the cell membrane is what this phospholipid bilayer is. Okay, that's the term we use to describe the structure of the cell membrane. really important term that you need to be aware of. It forms the phospholipid bilayer. And what that means is we've already looked at the structure. Okay, this is the hydrophilic head, hydrophilic, water-loving head. Why is it hydrophilic? This is the phosphate group. This is the top part. The circle is the top part of the phospholipid. So top part of phospholipid, and this sort of rectangle part is the bottom part. Now, as you can see over here, they line up, so they form this sort of bilayer membrane structure. Okay, so as you can see over here, this is the phospholipid bilayer. You've got the hydrophilic head, which interacts with the water. Okay, the hydrophilic head will interact with all water. So this is the phospholipid bilayer. So imagine you've got this structure in this square. Each of these lines in the square, you've got this structure. What happens is that water can interact over here. The water can interact over here because the phospholipids are water-loving. Now, the hydrophobic tail, they have to be aligned inwards inside over here because they don't like water. So if we imagine we had a membrane, yeah, this is the membrane, this square is the membrane. If we were to sprinkle some water on the membrane, what would happen is that the water would just like sort of stay on top of it. It wouldn't interact with these tails because these tails don't like water. They're hiding from the water. They don't want to interact with the water at all. Okay, that's why the tails are aligned inwards so they don't interact with the water. They absolutely hate the water. Okay, and because the outside interacts with the water, that's the hydrophilic head, the tails, they hate the water. The tails actually love lipids. Okay, because it's phospholipids. They're actually made up of fatty acids. So why wouldn't they like someone that's from the same category as them? So basically the hydrophobic tails, they love the lipids. So they allow lipid soluble substances to diffuse across the cell membrane. If I was to draw this a bit more sort of modeling, this is a really bad example, but I'm trying to. Okay, I'm trying to curve this around. I know it's not perfect, but imagine this as the cell membrane. Yeah, okay, and so on. Actually, I might just draw them all. Okay, obviously, this is just a rough shape. Okay, this is an example of a cell membrane. Okay, this square is basically this, but in a square shape. All the hydrophobic tails are aligned inwards, okay, because they don't want to touch the water, and all the phosphate heads are aligned outwards because they love to touch the water. If fats come in, fats can just go in. Fats are able to go in because these fatty acid tails, they love the lipid soluble substances. They love anything associated with fats. So fats can just go through straight through it. They're small enough, they can just go straight through. Water, however, will have to wait because water will have to be transported via channel protein or yeah, I think channel protein is the only one. And then you learn about aquaporins later in the spec. But basically, what is happening over here is only fats can move through. Small soluble fats can move through. Water cannot move through because these tails hate the water. Okay, and this phospholipid bilayer membrane structure, okay, or whatever you want to call it, just remember phospholipid bilayer, it's found in the cell membrane. That's what the cell membrane is made of, and that's what allows the cell membrane to move substances into and out of the cell by simple diffusion. Okay.